Acute inflammation. Acute inflammation is acute means it's fast. Inflammation is the way when these plasma proteins and fluids they jump from the blood vessel into extracellular space. That's the concept about acute inflammation. So why are we calling this acute? If we are calling this acute, it means we see something. What do we see? We see that if it's acute, it means it is neutrophil mediated. If it's chronic, it means it's lymphocytes mediated. We'll see an increase in neutrophils in acute. We'll see an increase in lymphocytes in chronic. So that's very important. So let's start reading it. What is inflammation? It's a process by which inflammatory cells, plasma proteins, fluid exit the blood vessel and enter into interstitial space. Explain acute inflammation. It's characterized by edema, characterized by neutrophils. In the presence of neutrophils defines acute. That's very important. Okay. Arises in response to infection or necrosis. Okay. That's very important because apoptosis never is followed by acute inflammation. It's never followed by acute inflammation. Necrosis is always followed by inflammation. Okay, what's the goal of acute inflammation? The goal is very clear: to remove the pathogen, to remove the necrotic debris. Now, if the debris are not removed properly, it can give rise to any sort of autoimmune disorders like SLE. We'll read about it. And acute inflammation, it's mediated by innate immunity. It's very quick, it's very acute. The only disadvantage of innate is that it's non-specific, means these are non-specific warriors, right? For example, if you got attacked by someone in acute, you were not prepared, what will happen? You will send whoever is present, right, to protect yourself. So it is innate, right? What is innate? Innate versus humoral. Innate versus humoral is that innate is neutrophil rich, mast cell rich, and complementary rich, right? Humoral is different T cell mediated. We'll talk about it. Okay, so we need something called as toll-like receptors. Now these are toll. Toll mean tax. So whenever you go inside any state or go inside any area, what happens that we have toll plaza, right? So these are toll-like receptors, and the only work of these receptors is to recognize. Okay, what are they recognizing? Let's see. Toll-like receptors, TLR receptors, are present on cells of innate immunity. Which cells? Neutrophils, complement proteins, these cells. Okay, so these toll-like receptors are present on immune system, macrophages, dendritic cells, right? Dendritic cells. Where are dendritic cells? CNS. So innate, these TLR are present on these cells, immune cells, and they are present on adaptive immune system. In innate system, the fast system, the TLR recognizes something called as PAMP. What is PAMP? PAMP is pathogen associated molecule patterns. It's a molecule, right? It's a pathogenic molecule, and it recognizes DAMP. What is DAMP? Damage associated molecule pattern, right? It recognizes, it's a recognition molecule. It recognizes it, and what happens is that once it recognizes it, it will recognize these bacteria, and it will recognize these molecules, and it will send these signal. Okay, let's start our innate immunity. So that's the work. So toll-like receptor, they recognize PAMP and DAMP. Example, CD14, example, aka TLR4 is a TLR receptor that are found on macrophage that recognizes lipopolysaccharide LPS, which is shared by gram-negative bacteria, right? So it recognizes this. Once you enter in a new state, in a new city, you go via toll plaza, and this toll plaza is the way. It's like, okay, someone has entered inside, so it recognizes it. Okay, that's how I remember it. Okay, so what chemicals is upregulated by TLR? Well, what does it upregulates? Something called as NFKB. NFKB is a molecular switch. It's a switch that turns on acute inflammation. It's a master regulator for both innate and adaptive immunity. Now, if you have any sort of deficiency in NFKB, NFKB, you will not have a good acute response to any sort of infection, right? So NFKB is a molecular switch. Other thing that they increase, upregulate, is called type 1 interferon. That's very important as well. So type 1 interferon, and what happens is that this type 1 interferon, it stops the viral replication in the cell. Okay, so it interferes, so that's why we call it interferon, right? And it's of two type, alpha and beta. Okay, we don't want to discuss right now because there'll be another different topic. Okay, so we understand TLR, it's a part of acute inflammation. We understand DAMP, PAMP, and LPS. Okay, LPS is shared by gram-negative bacteria, and DAMP and PAMP is recognized by toll-like receptors. Okay, another thing is NFKB is upregulated and type 1 interferon is upregulated. Okay, arachidonic acid metabolism. What happens is that, okay, let's read the question. Explain the production and metabolism of arachidonic acid. Well, arachidonic acid is made from phospholipase A2. Phospholipase A2 releases arachidonic acid from phospholipid cells. Now, this acid can be metabolized by something called as cycloogenase or 5-lipooxygenase. Okay, now these molecules called cycloogenase and 5-lipooxygenase, they produce something called as PG, prostaglandins, and something called as LT, that is leukotrienes. If we don't have arachidonic acid, we will not produce PG, we will not produce LT. If we have good amount of arachidonic acid, we will produce good amount of PG, we'll produce good amount of LT. That's the concept, okay? That's why we need arachidonic acid, because we need these mediator called prostaglandins and we need these mediator called leukotrienes. We'll know why do we need them, okay? What are the products of cyclooxygenase? What are their function? What is the pathway? Well, cyclooxygenase pathway produce something called as prostaglandins, PG. Now it makes something called as PGI2. It makes PGD2. It produces PGE2, right? So these products, prostaglandins I2, D2, E2, they increase something called as vascular permeability. They increase the fenestration. They increase permeability, means blood can go out and come out easily. It means the inflammation can, what is inflammation? Inflammation is when inflammatory cells can exit the blood vessel, right? So when the permeability is increased, the inflammation can happen smoothly. So permeability is increased, vasodilation is increased. Now I have written that vasodilation in red and permeability in blue because permeability increases mainly in venules and vasodilation mainly happen in arterioles. Now why it happens? Because arterioles have something called as smooth muscles at their epi layer, right? And at that epithelium layer they have smooth muscle, and what happens is that the capillaries, the venules, the capillaries lack smooth muscles. So only smooth muscle is responsible for vasodilation. So if a vessel has smooth muscle, it can vasodilate, it can change it, it can change its diameter. If it does not have any smooth muscle, it will not change the diameter. So that's why vasodilation happens at arterioles and permeability changes at fenestrided venules. Okay, again, another thing, PGE2 also mediates fever and pain, right? Note that vasodilation happens at arteriole levels and vascular permeability changes at post capillary venules. Okay, let's try to understand why it happens. So smooth muscle is present at epithelial layer, right? Capillaries lack these smooth muscles, so no vasodilation happen. Only permeability changes because of the fenestration. What is fenestration? These are the gaps between two endothelial cells, right? Endothelial cells. This is the gap, this is fenestration. And between these two fenestration, between this endothelial gap, we have something called as pericytes. It is a mural cell. Now this pericyte can, actually change the permeability of this post capillary venule. Okay, what are the products of lipooxygenase? So L stands for lipo, L stands for this leukotrienes. Okay, so lipogenase pathway, what are the function? 5 lipogenase pathway produce something called as leukotrienes. Leukotriene B4 attracts and activates neutrophils. Okay, so it is one of the very important, B4 is one of the very important chemoattractants for neutrophils. Leukotriene C4, D4, E4 mediate smooth muscle. It means it's mediating the vasodilation of arterioles, right? So it mediates, it means vasoconstriction can happen, vasodilation can happen. Okay, and vasoconstriction, bronchospasm, and increased vascular permeability. Contraction of pericytes open up. Contraction means these are getting smaller in size. Okay, contracting. So contraction of pericyte opens up space between two endothelial cells and fenestration gap increase. So that's the concept. Okay, you need pericytes for good fenestration. Fenestration happens at post capillary venules, and dilation changes at arteriole levels because of presence of smooth muscles. Okay. So we have B4, we have C4, D4, and E4. Okay, so what are the 4 important things that attract and activate neutrophils? Well, LTB4, we just read about it, leukotriene from lipooxygenase pathway. C5a, so there, what is C5 by the way? C5 means it's a part of complement protein, and C5 makes C5a and C5b. Now B goes in further pathway. A works for attracting these neutrophils. Okay, so now we know this is the part, right? What is IL-8? IL-8 is interleukin 8, right? So interleukin 8 mechanism by which macrophage call neutrophils, right? So they are calling, guns, big guns, that are neutrophils. So macrophages are calling IL-8. So IL-8 is the mediation between macrophages and neutrophils. Other thing, bacterial products such as, what are these bacterial products? This is one of the bacterial products, right? So bacterial products, once the macrophages can recognize these bacterial products, we can call the neutrophils via IL-8. We can use C5a, that is a part of complement protein, and we can use leukotriene B4. Okay, so leukotriene B4, what are the 4 important things? B4. Okay, 4, remember. TNF, tumor necrotic factor, and interleukin 1, based on Abbas book. I have written it because I read it. But these are more important topics. Not topics, these are more important. Okay. Mast cell. Not must, it's a mast cell. Mast cell, it means they do something. Why are they called mast cell? That's a good question. I will answer it in the next video. What are the 3 ways that mast cells get activated? Well, mast cells are found throughout the connective tissue. It means they are found on the mesenchymal layer. Okay, so they are found at mesenchymal layer, and they are activated by 3 ways. Tissue trauma, once you hit physically, tissue trauma can induce mast cell. Complement proteins such as 3A and 3A, right? 3A and 3A can, they can, so 3A is one of the early proteins of complement system. So 3A can happen. Cross-linking of IgE. Now, cross-linking of IgE, it means it's a IgE mediated hyperactivity. Cross-linking, I will tell you what cross-linking is. What happens is that these are 2 IgE together, right? These are 2 IgE, and this 2 IgE, what happens is that on a mast cell they are present, right? And they bind together. Okay, so if they bind together, this is called cross-linking. So cross-linking of cell surface of IgE by antigen, right? So cross-linking of cell surface IgE by an antigen. You need an antigen between them. I will tell you how it happens in this video of wait, I'll show you in the video of how reactive autoimmune system works. Wait, we'll talk about it. So just remember that cross-linking of IgE on by an antigen on a cell surface can start mast cell. Okay, this is I have drawn mast cell IgE. They are cross-linked by an antigen. You need something to cross-link people, right? What is acute response of mast cell? What happens when they are activated? First of all, they release something called as histamine, right? It's a neurotransmitter. It released which causes vasodilate. So vasodilate, it means the dilation, the arterioles diameter increases vascular permeability, post venular capillaries, fenestration, pericytes, right? Release something proteolytic enzymes. Proteo means protein, lytic means destroying. Proteolytic enzymes that kill bacteria and inactivate toxins. What is delayed response? Well, delayed response is after 4-5 hours of mast cell. What happens is that these will produce something called as AA, arachidonic acid metabolites. Metabolites means the AA has been metabolized, it has been digested, and what happens after digestion is that you have some, some waste material of the digestion. That metabolites is the digested. Now we need that metabolites for a lot of things, such as vasodilation and permeability as well, right? So AA metabolites are produced by mast cells, and leukotrienes are also released. So histamine, vasodilation, proteolytic enzymes, AA acid metabolites, and leukotrienes. L. What happens leukotrienes? Where does it come from? We know this by lipooxygenase. L stands for lipooxygenase, L stands for leukotrienes. Okay. Let's start about complement. What is complement? It is complementing an immune system, because that's why we call them complement. It circulate as inactive precursor cells. Once they are activated, they will complement the immune cell, right? What are the classic 3 ways of complement system activation? Classic pathway first, alternate pathway number 2, and mannose binding pathway number 3. What happens in classic pathway is that C1, I told you, binds to IgG or IgM, right? These are the first anti-immunoglobulin that are produced by naive B cells, right? So these cells, IgG and IgM, they bind with C1, and what happens that an antigen, which is, okay, let's read it again. C1 protein binds to IgG, IgM, which is bound to an antigen. So we have 3 things now. Antigen we have IgM, IgG, we have C1 protein. There are 3 things. Okay, once they are joined together, all of them, because you always need an antigen. Whom you're gonna make an immune response if you don't have an antigen? Antigen is always needed for activation, right? And IgG and IgM is also needed, immunoglobulin antibodies needed, and C1 protein. If you don't have C1 protein, what are you gonna activate? Which complement, right? So we need 3 things to activate: C1, IgG, IgM, and something called as antigen, right? Once they are activated, we call them activation of complement, right? So this is a classical pathway. It's classical because you guys have all 3 component of complement system, right? So this is why it's classical. Alternate, it means different one. What happens that we don't have antigen in this, the microbial products directly activate the complement system. So this antigen is not there, we have something called as microbial products. Okay, these product directly activate the complement system. We skip on binding of IgG and IgM, and we skip the binding of IgG or IgM with C1 protein. We skip that. So microbial products, they right away activate the complement system. So this is a fast pathway, it's really fast. Another one is mannose binding lectin. Now it is a pathway in which this MBL, mannose binding lectin, it binds to mannose sugar on a microorganism, right? So for example, this is a microorganism, right? It's really sad because he knows that he's gonna get destroyed. By whom? By something called as mannose. It has mannose receptors, right? So MBL ligand, it goes to this receptor on mannose, it binds and it activates complement. So mannose binding lectin, MBL pathway. MBL binds to mannose on microorganisms and it activates complement system. So we have 3 pathways: classical, alternate, and mannose. Classical is that when we have all three components of complement system, that is antigen plus IgG or IgM plus we have C1 protein. Okay. Alternate is that the products of microorganism direct right away, they bind and they not bind, they right away activate the complement system, right? They skip the whole three component shit. And the mannose binding pathway is that the mannose sugar are present on the organism. You need to have mannose sugar on the organism. If the organism lacks these, you know, mannose sugar, what will happen is that this mannose pathway will not occur. So mannose binding lectin pathway, right? The result of complement. What happens after the activation happens? How the membrane is attacked, how it's formed, the result of complement. Okay, so these have activated our pathway. What happens after this is that C1, we need something called a C1 convertase. Convertase means it converts, right? So C1 convertase converts sorry, not C1, what I'm saying. C3 convertase converts C3 into C3A and C3B, right? So C3A goes ahead into fighting, okay? It goes into fighting, and C3B goes further in the pathway, okay? So C3B convertase converts C5 into C5A. C5 convertase converts C5 to C5A and C5B, right? Now C5B joins with C6 and C9, right? And it makes membrane attack complex, something called as MAC, right? Which pokes the hole and kills it, right? It is very similar to how CD8 works. For example, CD8 also use perforins, and it produces granzymes, and it performs the killing of the cell, the bactericidal. So let's call it again C3, right? We need C3 convertase to convert C3 into C3A and C3B. Now we need C5 convertase to convert C5 into C5A into C5B. Now this C5B joins with C6, right, plus C9, right, to form something called as MAC. Now this MAC goes to the organism and pokes the hole in it. Once the hole is poked, the integrity of the organism is compromised, the structural integrity is uncompromised, and then the bacteria or the microorganism comes to a stop, comes to death. So C5B joins with C6 and C9 to make membrane attack complex. Okay, what is membrane attack complex? Again, C5B plus C6 plus C9 is membrane attack complex, MAC. That is very important to poke holes into the organism. Okay, let's talk about what are the key function of the complement proteins. These proteins I'm talking about, complement proteins, these are all complement proteins. So C5A and C3A, right? C5A and C3A, they activate mast cell, and we know mast cell can result in histamine, and histamine later can vasodilate or increase the permeability, right? So C3A and C5A activate mast cell. They stimulate degranulation. C5A is a chemotactic of neutrophil. C5A calls a lot of chemotactics, okay? C5A loves to call it, okay? C5A chemotactic. And C3B, right? C3B, C3 convertase converts C3 into C3A and C3B. So C3B is an opsonin, right, for phagocytosis. Opsonin tags the cell as a waste product for phagocytosis. If you have deficient in C3B, you will not produce phagocytosis. That can result in less complement reaction. MAC, the whole complex, MAC is membrane attack complex. It pokes hole in the cell and it kills it, right? So this was the concept about complement protein, right? This was the concept about histamine in mast cells, right? Let's talk about Hagman. Well, what is Hagman factor? H factor. We call this Hagman factor as factor 12 in coagulation cascade. Okay, factor 12 is called Hagman factor. What system does it activate? What activates, right? What does this Hagman does? Well, it's an pro-inflammatory. It means it induces inflammation, right? And Hagman is produced in liver, right? And on the exposure to subendothelial or tissue collagen during severe gram-negative sepsis, it is activated via subendothelial or tissue collagen in a gram-negative sepsis. Okay, it's a gram-negative. What else is in gram-negative? LPS is also presented gram-negative. It plays a very important role in something called a DIC, which is called disseminated intravascular coagulation. Okay, disseminated intravascular coagulation means it's a pathological activation of coagulation. Coagulation is happening. Coagulation, what is coagulation? Coagulation is just insoluble fibrinogen converts into, sorry, opposite. Soluble fibrinogen converts into insoluble fibrins. So that is coagulation. So pathological activation of coagulation cascade and severe gram-negative sepsis occur. So this is called as something called as disseminated intravascular coagulation. It activates complement proteins, right? This Hagman factor 12 activates complement protein. We know this complement protein is this C3 to C3AB, C5AB, then B makes B with the help of 6 and 9 makes MAC. This is complement protein. Again and again I'm telling this. It activates complement system, coagulation, and fibrinolytic system. Fibrinolytic means it destroys the fibrin. I told you coagulation is the process by which the soluble fibrinogen converts into insoluble fibrins. Now, if we can break this insoluble fibrin, we have something called as fibrinolytic substance. So, Hagman factor activates fibrinolytic system. This result in DIC, right, and kinin system. We'll talk about kinin system right away. What is kinin? Kinin system is a system in which this system cleavage, I mean it breaks a high molecular weight kinin into bradykinin. Okay, so kinin breaks into bradykinin. Okay, and bradykinin has histamine plus pain function in it. Okay, so we have a lot of medicine that are bradykinin inhibitors. Once you give this bradykinin inhibitors, you will not feel pain and will have less histamine production. Okay, and histamine is for vasodilation and vaso permeability increases, right? And it makes proteolytic enzymes. Okay. Okay. What are the two molecules mediated by pain? I told you inter prostaglandin E2. E pain fever, right? Fever E2. So PGE2, that is E2 and bradykinin. That's very important. What are the classical signs of inflammation? Well, first of all is rubor color. What is rubor? Rubor is redness, color is warmth. Now note that both of these things are mediated via one single pathway. What is this pathway? It's just vasodilation is happening. Once the vessels of arteries dilate, more blood can travel. Once more blood can travel, the skin turns red. The color turns red. Why is there is warmth? Because this oxygen rich blood is warm, right? So vasodilation, arterial relaxation by histamine. I know vasodilation, bradykinin again here, and PG, right? PGE2. Okay. What is the cause of tumor? Tumor means swelling. That's it. Why there is swelling happening? Because the permeability of post capillary venules, these venules permeability increase, the leaking of protein increase, right? Increased vascular permeability to post capillary venules mediated by histamine. I told you again, histamine, bradykinin. What is bradykinin? High weight kinin drops into bradykinin. That is bradykinin, right? Leukotrienes, LKE2 for fever, right? PG, prostaglandins, right? Tissue damage. Tissue damage can also, uh, attract neutrophils. So let's revise this again. What are the four important things that attract and activate neutrophils? That is leukotriene B4, C5 of the complement system, interleukin 8, right, and bacterial products. So that's very important. Okay. What causes dolor? That is a green, right? Dolor produces pain and fever, and bradykinin via high molecular weight kinin makes bradykinin. What causes fever? PGE2 causes fever. So macrophage releases something on this interleukin 1 and tumor necrotic factor. They go to perivascular cells of hypothalamus. We'll study about this. It's very simple. So this is hypothalamus on the perivascular area. Macrophages send is something called as interleukin cytokines, right? And tumor necrotic factor. It increases the activity of COX. What is COX? I think you know COX. I just told you what was COX, right? COX. Now COX causes increase in PGE2 and in the hypothalamus, which increases temperature. So it's COX mediated, okay? Interleukin 1. Let's talk about it again. COX mediated interleukin 1. Where is it? Here it is. Cyclooxygenase pathway. So cyclooxygenase pathway, this makes PG, prostaglandins, and 5-lipooxygenase pathway makes LT, right? So this prostaglandins PG is responsible for fever, right? So if we inhibit cyclogenase pathway, right? If we inhibit, we will not have fever because this COX pathway produces PGE2, and PGE2 goes to perivascular cells of hypothalamus, and then you have something called as fever. So this was the whole concept of acute inflammation. I hope it was not a very long video, but I wanted to make sure that we understand everything on a very conceptual basis, why everything is happening. So that was the whole video about acute inflammation.